feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Hi, and welcome to the National University of Singapore Yonglulin School of Medicine's Healthy Longevity webinar. Thanks for joining us. I'm coming to you tonight from central China. It's my first trip to China since 2019, and there's a lot of cool aging research going on here. So in the future, we'll have a few of the scientists in China that are doing some of the aging research that's exciting. Tonight, we have a tag team, two experts on the area of aging biomarkers, and I'll get back to them in a minute. Like always, I want to remind you to use the Q&A function to ask questions tonight. And we have Sharmila here who will be taking those questions and asking our speakers. But before we get started, I want to have Belinda, a research associate in the Center for Healthy Longevity, talk about her current study, Community Views Toward Health Span and Lifespan.
Thank you, Professor, for that kind introduction. Hello, everyone. As mentioned, I'm Belinda, a research associate here at the Center for Healthy Longevity. And today I'm thrilled to share with you our study exploring community fields towards health span and lifespan. It is no surprise that the global population has tripled between 1950 and 2020. Along with the surge in population, we also see a disproportionate increase in the number of individuals aged 70 years and older. This means people are now living longer, with a global average life expectancy of 73 years. However, these extended years may not be spent in good health, as many older adults struggle with chronic health conditions, such as diabetes and cardiovascular diseases. Therefore, we now see a gap of 9 years between lifespan, which is the number of years a person is alive, and health span, which is the number of years a person is alive while in good health. So to close that gap, healthy longevity medicine aims to optimize health span by targeting the aging processes across the lifespan. Through the use of artificial intelligence and biomarkers of aging, a person's biological age can be estimated. This then allows personalized health strategies for preventing chronic health conditions before they occur. So understanding where the public stands regarding this fast emerging field is a crucial step towards promoting healthy aging in the community, which brings me to the aim of our study: to understand the awareness, knowledge, and motivations of the general population towards health span, lifespan, and healthy longevity medicine. To do this, we conducted a focus group study. For those who may not know, a focus group is a qualitative research method that collects data through group discussions. In a typical focus group session, you have the researcher who asks the questions, and the responses provided by the participants are studied. So far for our study, we have conducted 13 one-hour sessions, each consisting of 1 to 7 participants. Our participants were adults currently living in Singapore, and given Singapore's rich ethnic diversity, we use purposive sampling, selecting participants across different age groups, genders, and ethnicities to gather insights from a wide range of populations. Participants answered a series of questions, some to understand their knowledge about health, lifespan, and health span, while others focused on their expectations about themselves, such as how long they would like to live and how long they expect to live. Additionally, we posed questions about their goals and motivations for staying healthy, as well as their thoughts and opinions about the Healthy Longevity Clinic. So currently, we are still in the middle of analyzing our data. So instead, I hope to share with you some insightful quotes from our participants. When asked what is health for you, many agreed that being healthy is being active, not only physically, but also mentally, and being positive always. So everything you do, you'll be happy also. However, some of them felt that if you need medicine for certain things, you are actually not in good health. You need medicine to prolong life, then it is not healthy life. You need to see doctors, get injection or transfusion, and all these things. So what's our next step? We aim to conduct a global survey to assess public knowledge and awareness about lifespan, health span, and healthy longevity medicine. And also our second aim is to investigate motivational factors towards health behaviors. By understanding the knowledge, awareness, and motivation, we can then identify key determinants or positive attitudes towards future health behaviors. We have already designed this survey, and we will conduct it in a total of 9 countries, starting with Singapore. So that's it from me. I would like to thank our PI and co-PIs of the study, and thank you very much for watching.
Belinda, we'll get back to you with more on the results of that survey. Everybody listening to this show is already an expert on health span and lifespan, so I know what you're going to say when you're asked those questions. So tonight we have something unique. We have 2 experts instead of 1. We have Jesse Poganik, who's an instructor at Brigham and Women's Hospital and Harvard Medical School, where he studies the fundamental nature of aging, and Mahdi Moqri, who's a joint researcher at Harvard Medical School and Stanford School of Medicine, where he studies epigenetic aging and its clinical translation. They both also serve on the executive committee of the Biomarkers of Ageing Consortium, and that's kind of why they're here tonight. They're going to give us a talk on something that's really near and dear to many of the listeners, the Biomarkers of Ageing Consortium. How do we characterize and validate biomarkers of aging? Jesse, I'll turn it over to you first.
Okay, thank you so much, Brian, and good evening to Singapore. Good morning from the US. As Brian mentioned, my name is Jesse Poganik. I'm an instructor in medicine at Brigham and Women's Hospital and Harvard Medical School. And what we'd like to talk to you today about is this recently formed Biomarkers of Aging Consortium, and how we really hope that this is going to become a key catalyst in advancing these really important tools, biomarkers of aging, to more clinical applications to allow us to broadly target healthy longevity across the world. But to tell you first about some of my personal research interest, I am really driven by this single question: what is aging? I think it's really the most important and interesting question in the field. And I would argue that we don't have a very good answer to it yet. This is also not a question that you wake up and go to the lab and answer. So we take a slightly different approach. We try to understand the nature of biological age. And Vadim Gladyshev, my PI, and I wrote an essay, which was published earlier this year, arguing for this approach. So what we try to do is we try to use these tools, biomarkers of aging, to understand features of biological age. For instance, we studied the temporal dynamics of biological age, particularly over short time periods. We try to understand the differences between how the biological age of the germline and the soma differ, because we know that these are very different types of cell lineages. Germline is essentially immortal, whereas the soma clearly ages. And so those differences can be informative in learning about biological age. And we're also interested in intertissue dynamics of biological age as we learn more and more about the similarities and differences between how different tissues and different organs age. And so all of these, we believe, will give us insights that will ultimately allow us to target the aging process and promote healthy longevity and extend health span and perhaps lifespan. And how we do all of this is biomarkers of aging. So let me give you one example of how this plays out. In a study we published earlier this year in Cell Metabolism, we tried to understand how biological age can fluctuate over short time periods. And we used a number of models of very severe physiological stress. And you see a summary graphically on top where we found that severe stress can transiently accelerate biological age, read out by these biomarkers, but after a period of recovery, it can relax down to the baseline. And here's just one example out of several models that we examined in this paper, that of major surgery. So this was from a group of elderly individuals who had traumatic hip fractures and needed emergency surgery to repair that fracture. You can see that from pre-surgery to post-surgery, there was this significant increase in biological age read out by this biomarker called grim age. But when the patients were discharged from the hospital and their blood was tested again, they were back down to their baseline biological age. And so this tells us something about the effect of severe stress, but more importantly, it tells us a fundamental feature of the nature of biological age, that we believe that it can be fluctuational over short time periods. And we observe this at many different levels, not only at the level of DNA methylation, but also at the level of gene expression and metabolites. So we believe that this is a true effect on biological age. But what I really hope to highlight here is the power of using these biomarkers, because for the first time, these biomarkers, even in a preclinical context, such as this study, allow us to quantitatively assess aging. And so biomarkers of aging have really become a central feature in the field. And they've been rightly, in my opinion, called a revolution in the field, because again, they for the first time allow us to quantify the aging process. So we've known for nearly 100 years that aging can be modifiable, and I certainly don't need to convince this audience of that. But really, it's only within the last decade or so that we've had this explosion in the development of biomarkers of aging, all shown here in blue. And in spite of this really rapid progression of this space, there's this strange contrast that goes on, because the reality is that we are constantly applying these biomarkers in epidemiological context to understand how various lifestyles, genetic backgrounds, et cetera, affect biomarkers of aging. We're applying them in preclinical basic research. I've just shown you an example of that to study things like anti-aging and cellular rejuvenation. And what's interesting is that we're also applying them in clinical trials, both those that have been completed, those that are ongoing, and those that are planned. But in spite of the fact that the field has really taken off, there are very basic questions that people still ask. So, and this is just to reinforce this idea that these biomarkers are being used in clinical trials. This is a snapshot that we put together of the for our paper, which I'll tell you about in just a second, showing all of the clinical trials. But we still hear these very basic questions all the time. What biomarkers should I use? What do different biomarkers measure? And how can we validate biomarkers and ultimately progress towards regulatory approval? Because if we want these biomarkers to be broadly beneficial to society and to the world, ultimately regulatory approval is where we need to get. And so recognizing these unmet needs, the really critical needs to answer these questions, we formed this Biomarkers of Aging Consortium last year. And I think we're coming up on our 1-year anniversary very soon. And I would say that really our greatest strength as a consortium is the community of experts we've put together. You see here on the slide our steering committee made up of 5 really top scientists of very diverse expertise. Vadim Gladyshev from Harvard, Andrea Meyer, who needs no introduction to this audience, Vittorio Sebastiano, Mike Snyder, and Eric Verdon. And on the bottom, you see our executive committee that's kind of more in charge of the on-the-ground aspects of the consortium. Mahdi, who you'll hear from in a minute, myself, Dane Goebel from the Methuselah Foundation, and Alison Duetman from the Foresight Institute. And we have complemented now our steering and executive committees with a scientific advisory board composed of Luigi Ferrucci. He's the national director of he's the director of the National Institute on Aging in the US. I don't know if anyone's heard of Brian Kennedy, but he's a really great guy. We're very lucky to have him. And we also have Steve Cummings, an expert in more clinical aspects. Steve Horvath, who you may probably know is one of the real pioneers of this biomarker field, particularly in the realm of DNA methylation. And Jamie Justice, who's had a very long career thinking about clinical applications and regulatory approval of these biomarkers. So we really benefit from the broad expertise of this top-notch group that we've put together. And since we conceived of this idea in September, and I really should say at this point also that Mahdi was the key driving force in bringing this community together. But since we've conceived of this idea, we've produced our first work, which was on trying to establish some baseline of consensus across concepts related to biomarkers of aging. I'm going to tell you more about that in just a second. We have a second work which has been submitted for publication about cross-population validation of these biomarkers. Mahdi is going to tell you about that after I'm done talking. We have an in-person symposium planned where we're going to bring our community together to talk about this. That's coming up in just about a month. And tied to this, we're working on a third work, which is to leverage our panels that we're bringing together for the symposium to define the critical next steps for the field. So the way we go about our consensus building work is that we identify panels of experts. This is the panel we put together for our first manuscript. And you can see that, first of all, these are the leaders in the field. And they really have diverse expertise going from very detailed preclinical basic research studies to almost exclusively clinical focus to also regulatory experts. And we leveraged this panel of experts using a Delphi-based method of consensus building where we had a core drafting team that was composed of me, Mahdi, and Chiara Herzog, a postdoc from Innsbruck University. And so we together drafted a first draft of this manuscript, and then we went through iterative rounds of soliciting feedback from our expert panels. And in the end, through this process of back and forth, we were able to achieve some level of consensus on really critical concepts. And this resulted a few months ago in the publication of this perspective in Cell, which is the inaugural work of our consortium. And the point of this paper was really to kind of level set for the field, define some key terms, propose a classification scheme for biomarkers, and talk about some of the key considerations that we need to be thinking about. So we were able to establish some working definitions. We need these working definitions to be able to communicate with each other. It's not to say that these definitions will ever change, but for now, we were able to achieve this. So for instance, we've defined aging as the process of accumulation of consequences of life, such as molecular and cellular damage that leads to functional decline, chronic diseases, and ultimately mortality. You can really see the kind of consensus building that went on in this process because this covers everything from very molecular considerations like damage to very clinical manifestations like disease and mortality. We were also able to propose this sort of two-part definition of biological age, where we deal with the conceptual side of it, which is an individual's age defined by the level of age-dependent biological changes at the cellular and molecular level. And we also give a practical definition where we explain how most people consider biological age a number matching the average chronological age in a reference population. So these working definitions were really kind of, in many ways, the first step to being able to define any of the rest of the critical topics we discuss in this paper. So we broke down various biomarkers of aging into different categories because there are many of these being developed all the time. We broadly categorized these as molecular biomarkers. This is the largest group in this space right now, which can be divided further into omics approaches, things like DNA methylation, or specific molecules. And within omics approaches, you can also have biomarkers that are working at the level of DNA methylation, gene expression, proteomics, or metabolomics. There are also physiological biomarkers. These might be things that you've seen at the clinic that deal with functional performance or physical characteristics. And more, we're seeing the development of biomarkers based on digital technology like wearables and also non-wearables. So this allowed us to break these down into these different categories. And then we were also able to conceptually separate these biomarkers. So biomarkers can fall into these different categories you see here. They can be predictive or prognostic, meaning that they predict a future outcome. They can measure a response to an intervention, or they can be used as discovery biomarkers. You can leverage the result of biomarkers to try to discover new interventions to target healthy longevity. And you can see here the different use cases for these biomarkers where, again, predictive biomarkers can predict a change in health outcome, response biomarkers can measure the effect of an intervention, and so forth. So all of these are being constantly developed, and we've tried to catalog them here because different biomarkers are useful for different applications. And the reality is we probably need a broad suite of these to really target healthy aging, because aging, of course, is not just one thing, manifests at many different levels. And finally, we were able to discuss the evaluation of biomarkers of aging, and we break them down into these categories you see here. And these really run the gamut from things such as feasibility to response criteria for interventions, and even things like cost considerations, which become very important for these omic biomarkers, where the tests themselves cost a lot of money to run. And we discussed this idea about how geroprotectors, which are interventions that can promote healthy aging, can inform the development of biomarkers of aging and vice versa. So our next steps are to have our symposium. This is coming up in December. And once again, we're continuing this theme of really being very lucky to benefit from a really broad expertise of the panelists that we've invited. This is taking place at the Buck Institute on December 4th. And in addition to this in-person event, we're also leveraging this panel to write another manuscript. This just gives you a flavor of this manuscript. It's based on surveying these individuals. Here, just an example of 2 questions we're asking them. And this work is being spearheaded by Chiara and Ludger, who's a member of our lab as well. So this is really going to allow us to define the priorities for our next year of work and kind of set the stage for our next steps. So in summary, what I've told you is that we've assembled this really excellent group of experts. I can't emphasize enough how lucky we are to have so much willing cooperation among these experts of really broad expertise across the aging biomarkers space. And I think you can see from our inaugural work, and I hope you will continue to see in the months to come, that this expertise is allowing us to take concrete steps to advance biomarkers of aging. Our immediate next goals are to survey our experts to define the next steps for us. And also, we have a particular interest in systematic validation of biomarkers, which is really a key priority for these tools. And this is what Mahdi is going to tell you about. So I've acknowledged the consortium along the way, and I don't want to take too much of Mahdi's time, so I just wanted to highlight a few more people who have been really key to our work. And thank you very much to our sponsors. And now I will turn it over to Mahdi.
Thank you very much, Jesse. Great talk and right on time. Let me share my screen. Thank you, Brian, for inviting me. I'm going to continue talking about our work with Biomarkers of Aging Consortium. I'm a Jones Fellow in Aging at the Gladyshev Lab at Harvard and the Snyder Lab at Stanford. Just a bit about my background. I'm an informatician by training. And before joining the field of aging, I was building predictive models for patients' outcome. And it was amazing how age showed up as the most predictive, as the strongest predictive factors in various settings for Alzheimer patients, for ICU patients, for COVID patients. And it just, I got just so curious that I decided to leave my faculty position and for a sabbatical to go to Stanford, work with Dr. Snyder and Dr. Sebastiano to learn more about aging and how we can better measure it, and later join the Gladyshev lab to understand aging and how its biology. So my work with the Snyder lab is mostly on this longitudinal cohort called IPOP. It's a longitudinal cohort of people deeply measured across more than 10 years now. And we have all these different omics for these people. And recently we published a paper studying all the lipid species of these people longitudinally measured and how that changes with age. More recently, we are studying their whole genome epigenetic in collaboration with Biomodel. We also invented a new method to study the epigene of these people at a whole genome level, which is now being patented at Stanford. I'll talk about this method that relates to the causal understanding of biomarkers in a bit. And at the Gladyshev lab, my work is more about understanding the biology of aging and how that connects, how that links to these biomarkers. Our work on causal epigenetic aging is forthcoming, and we also have a work on comprehensive study of different biomarkers. But the one that I want to mention in the context of consortium is our framework for validation of biomarkers of aging. If you just Google for biomarkers of aging, you can see all sorts of products out there that is currently sold directly to consumer or clinicians, and you can just purchase them, do this test at home, and get a result that represents that's supposed to represent your biological aging. But the questions that we get very often is how to interpret these results. What if is my number higher or lower than my actual age? And the other question is, especially from clinicians, which one of these biomarkers should we use? Should we use omic biomarkers first of all? And if omic, which one of them, metabolomics or epigenomics? And now there are so many of them out there that we need a better understanding of them. So my research, my basic research and my work with the consortium focuses on addressing these two questions, building better informative and interpretable biomarkers of aging, and with the consortium to benchmark these biomarkers. And we are also hosting a competition that I'll mention about in my last slide. Right now, the way we use these biomarkers in our labs, preclinical studies, is we have these rejuvenating or anti-aging interventions, and we measure these biomarkers before and after, or we measure these biomarkers in young, old, and old treated groups. And these biomarkers gives us some feedback, some insight about what's happening at the molecular level. And it's very common now. It's performed in almost every aging and longevity lab who are doing preclinical studies. But still, there are some open questions about these biomarkers itself that we need to better answer. To give you an example, the epigenetic biomarkers, which is the basis of most existing, most current biomarkers out there that you can buy, are based on DNA methylation of individual genes that correlate very highly with age. And if you put enough of them together, you can build these clocks that very nicely move with age. And for some people, it happens to be higher than their chronological age, and for other people, lower. And that difference correlates, also can predict your aging outcome. And now it's very well established that this accelerated aging very nicely predicts different aging outcomes. And this is the basis of most existing biomarkers, but it's very hard to interpret and make an action based of them. So if I know this, what can I do to improve it? And what is causing my accelerating aging, for example? So that incentivized us to build a more biologically driven and informed model. For example, with Dr. Sebastiano, we put together a team of experts in epigenetic aging, in characterizing epigenetic aging, and we found different features that are linked with age and with biological age. And using these feature-based models and machine learning, we found this specific feature of binding of polycomb repressors that nicely correlates with age and also some aging outcomes. And that was an example of an interpretable model that now we have, and it uses the whole genome, and we can make sense of what it means if it's higher or lower, and how to tie that to the biology of aging. And regarding our work with the consortium, after our first work, the consensus building and understanding the broad challenges in the field, one of the highlight of what we learned is we need more validation. We need cross-population validation of existing and new biomarkers to be able to talk to regulatory and to show them that these biomarkers are generalizable, they are consistent. And together with the same team of the consortium and inviting more people with expertise in population studies, we put together a framework for how to validate a biomarker and how to do a cross-population validation of these studies. And the idea is we want to move away from validating or testing one biomarker in one population to comparing and benchmarking different biomarkers in a population, and then comparing multiple biomarkers in diverse populations. So first of all, we can compare, we can see how they do compared to other modalities of biomarkers, and then we can make sure that they are consistent across diverse background of populations. And we also put some stepwise process on how to validate these particularly composite or omic biomarkers for the field. This work is still under review, but hopefully coming out. But what we learned from this work was, first of all, there are already a good number of existing biomarkers that have been validated in different levels. And some of them are really promising. Some of them are validated in large cohorts of more than 10,000 people, or very diverse population of multiple cohorts from different backgrounds. And some are actually reporting very high predictive power, which is at the level that we can, that are very clinically relevant. And just to give you a context about these performances, this is one particular epigenetic clock that is reported to have very high predictive power, and it can stratify the population into groups. And if you are in the high top group, after more than 10 years, only a few percentage of these subgroup pass away, and in the lower group, more than almost half of them die after around 10 years. So the difference, the gap is very clinically relevant. And but what we need for this study is more validation in more diverse population. And more recently, last few years, we see that independent groups have been comparing and benchmarking these studies in one particular cohort typically. And what we need is to move this forward and compare these and benchmark these different biomarkers across population and larger cohorts. And to help with this process, we put together an open source library. It's called BioLearn. And we started to standardize and harmonize the existing biomarkers, starting with the epigenetic ones that are more popular. And now with this open source library, you can apply any biomarker that's publicly available on any data using a one-line command, using a very short interactive command. And these biomarkers, and same thing for clinical. Now we are building on the clinical side of it, that in a standard way, it produces those measure of predictive performances. So it's now reliable and replicative, because before this, people had to start the whole thing from scratch, and there are some assumptions in these models, and there are some pre-processing of the data that affects the result of model. So this is our first step toward standardizing the process of validation. And at the same time, we are also hosting a competition, a fair way to benchmark and test these biomarkers using samples from a biobank. We have collected 500 blood samples and assayed them with the most recent technology for epigenetic analysis. And we are making all this data publicly available. And this data will be used in 3 phases of competition to predict the age of donors and the time to survival for those who passed away, or mortality outcome. And the last phase would be the multimorbidity and other aging outcomes. It's a very well balanced population of sex divided and nice diverse background. And this is an ongoing study. We have access to all their medical records and fully consented donors. And this, we hope that this will be a resource for the field, similar to other fields, to benchmark their method and every year to update their performance and hopefully improve these predictive performances. And this will be launched at our symposium next month, December 4th, at the Buck Institute. And it will be a year-long competition, and every phase will be a few months. We are still planning the details with the help of the Methuselah Foundation. And but our goal is to launch this competition on December 4th for the field. If you are interested to learn more about our work, we have a website that's very active. And there are also other videos that we prepared for the community that has more details about our activities, and you can find all of them online. And I also want to thank the Gladyshev Lab, Snyder Lab, and Sebastiano Lab that are supporting these works, and Methuselah Foundation, and all the audience. And in particular, I want to thank Jesse, the co-director of the consortium, and this work wouldn't be possible without his help.
Thanks, guys. I think that's very interesting. It brings up a lot of questions, and I'll try to keep it relatively short so we have some time for questions from the audience. So feel free to use the Q&A button. I'm sure there's some thoughts out there from all you listeners. I wanted to start with that paper you published, Jesse, on the traumatic experiences accelerating biologic age. I have a personal interest in this. I have a number of traumatic surgeries, so I'm kind of curious. You read the literature, most of which is from years ago, showing that people that go through traumatic events have a shorter life expectancy. And yet, in your studies, you're showing that people that go through these events have an accelerated biologic age, but then it returns back to some norm. Does it go all the way back to where they started from, or is there some residual effect from these traumatic? And it's not just surgery. I know you looked at some other things as well.
Yeah. I think this is a really interesting question, and I'm going to have to disappoint you by telling you that I don't have a good answer for it. But I think it's very important direction for the future of that work to understand how these transient accelerations feed into the overall trajectory of biological age and lifespan. We don't understand that, in a thorough way right now. We were relatively limited, actually, in what we were able to examine, because very few people measure these omics data sets over short time periods, and fewer still then track years later what happens to the individuals. What I can tell you, though, is that while we do see on average across the cohorts that we examined that the age goes back to the baseline, we don't see it always. Even in that one slide that I showed you, you may have noticed there was one individual who was elevated after the surgery and then kind of stayed at that level. So one thing we would like to do, and we plan to do, but is very challenging from a data point of view, is to understand whether the people who have, say, an impairment in their recovery of these biomarkers also have poorer clinical outcomes. Again, I'm sorry to disappoint. It's not something we've done yet, but I hope to have an answer for you someday, because I think it's very important if these fluctuations on the short term feed into the overall trajectory of aging.
Well, and I think the studies that we've been doing and others as well have shown that even in less traumatic experiences, there's some fluctuation with biologic age. And, that's been controversial to some people in the field. I think there was a perception that age can't fluctuate around, it's going up and not up and down. And I'm wondering, what that means. And one way to think about it is that biologic age is fluctuating. And when you're at a higher biologic age, maybe at that point you're more susceptible to other events that happen that then sort of lock you into that higher biologic age. So um, maybe it's a measure of relative risk, this biologic age, a little bit. What do you think about that?
Yeah, absolutely. I agree. And I mean, one thing I would say is that one interesting example in this regard is COVID-19. So one of the models we studied in that paper was very severe COVID-19, where we've shown that in general, people's age is transiently accelerated by very severe occurrences of the disease. And we know now also that there are some, at least initially, successful clinical trials targeting aging pathways to try to treat COVID-19. So that's one little hint, I think, at where we're going. But I would say the broader point, to bring it back to the consortium and biomarkers in general, is that this is a really a key question for this field. We don't understand the extent to which biological age or these biomarkers of biological age fluctuate as a function of, anything, everyday life, mild illness, severe stress. We have this little glimpse from this initial study, but there's a lot of work still to be done to understand this, because it becomes a very practical question in my mind, because you need to know when these biomarkers should be measured so that you're not fooled by something that may be transiently accelerating them and may or may not be relevant to aging. So, I think both things are very important. Number one, understanding how these biomarkers and biological age behaves, and then also understanding the connections to the underlying biology and, ultimately risk to health.
Maybe I'll take your lead, because, I can talk about science, the deep science, forever, but, we're here talking about the consortium as well. And so you both mentioned path to regulatory approval. And I think our listeners, when you talk about drug approval, they have some awareness of what it takes to get a drug approved. But how do you get to regulatory approval for biomarker? And how do you see that playing out for aging biomarkers?
Mahdi, do you want to start? Because I've been talking a lot.
Yeah, of course. It's still very early. There's a good model for us, the Biomarkers Consortium, which is a collaboration between NIH and FDA. And they have been working on biomarkers of Alzheimer's, biomarkers of other diseases. So one approach to our work could be following the lead of biomarkers of diseases. So there are a lot in commonality. So, for example, the frameworks that we presented in the Cell paper are borrowed from the NIH framework for, it's called the BEST framework for biomarkers. And there are a lot of commonalities. But then the challenge would be how to adopt these for aging, which is not considered a disease. So there is the caveat of customizing those models for qualifying a biomarker for aging. But there's a well-defined process for qualification of biomarker. And the first step is to have a consensus, a scientific consensus about their context of use, their predictive performance, or their general utility. So that's what we are also starting with, driving the consensus in the field.
One way to think about it, and I'm trying to make an analogous thought process to what goes on with interventions. Some people like us were using biomarkers as outcomes, and that's a bit circular when you're trying to validate the biomarker. But if you look at something like the TAME trial, they're trying to prevent multi-disease simultaneously. And so can these biomarkers be measurements of onset of multiple diseases? And I guess that's where some of the biomarkers have gone more recently, predicting morbidity or multi-morbidity. So is that a path toward regulatory approval potentially?
Yeah, I think it could be. I think it could be. And, part of, so I would say that, in the kind of broad scheme of omics biomarkers, we kind of started with prediction of chronological age in the earliest days. And many biomarkers are still primarily trained on chronological age. And then kind of the next big general wave was prediction of mortality risk. This was things when this was grim age, for instance, and related biomarkers. And now I think the interest is in predicting diseases of aging or aging-associated diseases. And so, I think we will see more development in that space, and it could well lead into regulatory pathways, especially because those are primarily conceived from the standpoint of disease, as Mahdi said. But I would also just point out this is kind of exactly how we're thinking about this competition that we're running. So we have 3 tracks that we're planning that are laid out exactly as I just described, essentially. The first to predict chronological age, the second to predict mortality risk, and then the third, and we predict the most challenging, is prediction of multimorbidity or occurrence of aging-associated disease. So I think, Mahdi can feed in because he's kind of more involved on the development side of biomarkers. I think that we'll see more work in this direction. I think that's where kind of the appetite, if you will, is. And it's kind of, I think, the next big thing for aging biomarkers.
Maybe I'll ask one more question, and this is kind of a philosophical question. It's one that I kind of grapple with in these kinds of projects. And, I think these projects are really important to bring the field together, but what does consensus really mean? I mean, you have a field that's emerging and people with all kinds of different thoughts. And I think the challenge, I'll start with my answer and you can tell me that I'm right or wrong. I think the challenge is encouraging people to still maintain that creativity and go in different directions, and yet have some kind of common ground that people can communicate with. This is a perfect example of that because all of these clocks are within 15 years of age. And so, how did you approach the idea of consensus when you started this project?
So maybe I'll start, and Jesse also might have something to add because we have this discussion ongoing in an ongoing way. So for us in the lab, every once in a while, maybe once in a month, we have these questions of what is aging. And if we don't have a good definition or a working definition for the purpose of use, it's really hard to continue that discussion because people are talking about different things when they are using the same words. So, for example, there are now more than 300 different theories of aging, and every one of them is proposing different definitions. And a lot of them have some points, but for the purpose of biomarkers of aging, we need a specific working definition of aging, biological aging, and biomarkers of aging. And that's the part that we needed a consensus to be able to work on the practical sides of it, talking the same language and using the same words and having the same understanding. Jesse, do you have anything to add to that?
I would simply reiterate that the point was to achieve kind of a baseline of consensus. It was not. It's not to say that we want to establish dogma for the field. You must do this. You must do that. You must consider things this way. Even the definitions in the paper, we emphasize these are working definitions. They're tools that we need to progress forward, as Mahdi says, to speak the same language in terms that we can basically agree on. So we think a lot about trying to keep the consortium as unbiased and inclusive as possible. And it's for exactly the reason that Brian mentioned, that we don't want to lock people into any specific way of thinking. We don't want to stifle innovation in this space. I don't think we're going to, because I think this space is really vibrant and there's a lot of amazing science going on. But the goal for when we say consensus building was to achieve a baseline of consensus that we can build upon. It's not to try to restrict innovation.
Hi, everyone. I'm Shamila, the research associate from Brian's lab, the Center of Longevity. Dr. Jess, I have a direct question from you from the audience. So they refer to your slide BA. They said if BA is reversible, increased by physiological stress, how about the thermodynamical component of aging? And what is the link to the reprogramming then? That's a question.
Yeah, and they're also asking about Peter Fedichov's work. So I would say, if you're familiar with Peter Fedichov's work, it's actually a nice complement. So in that work, they've studied biomarkers that were based on the results of clinical blood tests. And in our work, we primarily focused on omics biomarkers, mainly DNA methylation biomarkers. And I would say broadly, the results agree. In as much as they show that the biological age read out by these different methods is fluxional, and this idea of returning to baseline may contribute to kind of the overall lifespan trajectory of biological age. What is the connection to reprogramming? Honestly, I have no idea. We haven't connected the two concepts. And reprogramming and aging, I think you could have a whole separate webinar series about understanding the relationship between the two things. So unfortunately, I don't have a good answer for that part of it.
Okay.
This is Brian. First of all, we've had Peter on a previous show, so if you want to know more about that, you can go back and look at the YouTube video a lot. We've also had Vadim and a lot of the other people discuss tonight. Second of all, I'm sort of semi-functional here, so I'll let Sharmila go ahead and ask questions. But I just want to ask Joe if you can, I can't see the clock, so if you can text me when we're, about 2 minutes from the end, that'd be great. Thank you.
Thank you, Brian. Understand telomeres reside at the end of chromosome plays a significant role in aging. Are these telomeres be considered as biomarkers of aging?
Maybe I can take that. So is this still an active area of research? We see still very good quality research on telomeres and their length and how that connects to aging, and interestingly, how it connects to other biomarkers of aging. For example, there is now a proxy measure of telomeres based on epigenetic measures. So it's still an open question in the field. But one reason that a lot of researchers, a lot of groups have focused more on other omics compared to telomeres is simply the informatics aspects of it. The number of measurements that we get from telomeres is in the order of tens per cell, right? How many telomeres we have in a cell. And that's a limited amount of information. And it's also representing one form of damage or aging, cellular aging. But on the other side, for example, for epigenetic measurements, we get 30 million measurements per cell. And a lot of them could be actually used to almost completely estimate the telomere length in addition to many other things. So one reason that maybe the field is focusing more on omics compared to molecular, single molecule, or even telomeres and other classic measures of aging is just simply higher resolution of measurements.
Okay, they also asked that how did you choose which biomarkers are related to aging? Which one of these are related to Alzheimer or other diseases? How if the biomarker is related to other diseases? So it means it's not really specific.
I have a short answer for that, and if Jesse wants to add to that. So one criteria for biomarkers of aging is it should be predictive of diverse aging outcomes. And the next step would be the causal or in the causal pathway. But the key here is that if a biomarker only predicts the outcome of cancer, that's a biomarker of cancer, or for Alzheimer, the same. But we consider a biomarker a good biomarker of aging if it can relate to diverse aging outcomes. For example, the biomarkers that I showed on my slides can predict mortality from cancer with the same accuracy of mortality from Alzheimer, and we know they have different biology. As much as we know, it seems it's capturing the broader aspects of cellular aging.
Okay. So do you have any opinion on having a biomarker which can be used every day by everyone?
That's a tough one. I'll let Jesse to answer that.
My answer is please have us back in a year or so. And once we complete our validation work, we may be able to give you an answer. No promises. But right now, we need to do more validation, I think, before we can make any recommendations.
They also
Maybe I can jump in with a question because I can't resist, Sharmila.
Sure.
So, you talk about clocks made from one type of omics or one type of measures. What about sort of multi-omic clocks? Have those been generated yet? And do you think that there's going to be more information in those than there is in a deep single omics data set?
That's a question for Mahdi.
Yeah. So this year alone, we've seen a few good multi-omics works. One of them from our own biobank, from the MGB biobank, Mass General Hospital biobank, and our colleague, Dr. Lasky Su group in collaboration with other companies. But there are a few, and one of them from your own, Andrew Meyer, using UK biobank. There are multiple good quality research performed on multi-omics in addition to Michael Snyder's group work. So I see all of them as promising, and they are finding new insight. And it's just, to me, it's reasonable to assume that combining different modalities of data would produce higher quality outcome. And if I have to predict, I would bet that the future biomarkers will be multi-modality. Yeah.
Okay, I need to jump in here. Thank you, Sharmila. I didn't get a chance to introduce you because of the computer crash, but thanks for joining the show and asking the questions. Thanks, Jesse and Mahdi. We'll have you back in a year, and you can tell us everything that's fully validated at that point. I want to remind people to use the chat function and panelists and all attendees option to leave comments and feedbacks on the show. Please sign up for our Unlock Healthy Longevity Supplements Conference. There may be an image up there for that. I can't tell from my connection. It'll be February 29th and March 1st, 2024. We have a great list of speakers, and I think that this is one of the first meetings of its kind, looking specifically at supplements as geroprotective interventions. So don't miss that. The Center for Healthy Longevity is looking for a postdoc in geroscience, clinical trials, nutrition supplements to optimize the health of middle-aged individuals and to work with a dynamic team here at NUS. Please look for the link and contact us if you're interested. That'll be in the chat box. Our next show will be November 9th, 2023. And we'll have David Gems. For those of you who know David Gems, I guarantee you it'll be a provocative discussion. His title is going to be Bad Spandrels, Biological Constraint as a Cause of Aging. I'll be with you next week, and we're going to leave you tonight with a little bit of humor, hopefully, talking about how every generation thinks they're the most progressive. Thanks a lot.
We think we're the most tolerant generation as well. Oh, compared to our grandparents. Our grandparents are so racist and sexist compared to us. Wait till we're grandparents and our tolerance has run out. And your granddaughter brings home a robot. No robots in this house! I don't trust them! Not after World War III! Granddad, don't say that! I'm gonna marry him! You don't marry a robot! It's not natural. Marriage is between a man and a woman, or a man and a man, or a woman and a woman, or a non-binary gendered person, or a transgender person, a queer gender person, or a trigender person, or a bigender person that doesn't identify as a gender, a bisexual, or a asexual, pansexual, my mate who identifies as an alien. But no robots. You don't f*** Siri. Don't call them Siris, Grandad, that's offensive. They're all Siris to me, sweetheart. Thank you very much.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.